Thank you for joining us today to learn more about a new kit that we will be launching very soon, which is for simple, fast, elegant, directional RNA-Seq library preparation that provides a seamless depletion of ribosomal RNA and globin mRNAs. So I'm Dave Schuster. I'm the head of R&D for Quanta Bio. And again, thank you for taking the time and joining us today. So for today, what I'd like to first do is give you a brief background on Quanta Bio, and then jump into the specifics about our new kit, the SparQ RNA-Seq HMR Kit. So Quanta Bio was founded in 2001 as Quanta Biosciences by a small group of us from what I still refer to as the original Life Technologies. This included senior R&D scientists such as myself, a business executive, and experts from other areas of the company, including biomanufacturing and product management. If you might not be immediately familiar with Quanta Bio, but this team, while at Life, played key roles in the development of products such as Platinum Tac and the superscript RT-PCR, qPCR kits. You may be more familiar with those. And when founding the company, our goal was really, we leveraged this expertise and experience that we brought in the fields of biochemistry, enzymology, and molecular biology, with the goal of providing both innovative, highly robust products that simplified complex workflows for reverse transcriptases, cDNA synthesis, and application to PCR, real-time quantitative PCR. We leverage our experience in these fields of biochemistry, enzymology, and molecular biology with the goal of providing innovative, highly robust products that simplify complex workflows for reverse transcription, for RT-PCR, qPCR, and so forth. And we were responsible for the development, production of Bio-Rad's iScript and IQ and ITAC super mixes until about 2007. I think probably we're most best known for these days is really our ToughMix product line. And we've been a leading supplier of one-step RT-qPCR reagents for COVID testing throughout this pandemic. More recently, we've directed our efforts towards simplifying and streamlining workflows for next-gen sequencing and providing products that both reduce intrinsic technical errors and deliver reliable and reproducible results. The focus here is so that, not that we can necessarily eliminate requirements for proper controls. However, our hope is to enable you to be able to address and focus on the relevant issues of your research area and not be mired in technical nuances of supporting methodologies. So we've had a complete product line for DNA NGS library preparation for quite some time. This includes, if you're starting with Covaris fragmented DNA, so the SparQ DNA library prep kit carries out end polishing, end repair, ligation, and then carries it through the rest of the workflow. The SparQ frag and library prep kit includes an enzymatic fragmentation module that is tunable, that then and adjustable to a variety of input and quality of different qualities of DNA to provide a highly reliable enzymatically fragmented DNA to take you through the DNA library prep process. And it's supported by a robust hi-fi PCR master mix, the SparQ UDI adapters, SparQ PureMag beads, as well as a universal library prep kit. Now, what we're going to focus on today, though, is we're bringing in new technologies for total RNA-Seq. This RNA-Seq kit includes depletion modules for human, mouse, rat, ribosomal, and globin sequences. And again, it can be married in with the SparQ UDI adapters, PureMag beads, to, so that we can provide a complete solutions for both DNA and RNA library preps to feed NGS workflows. So why study RNA-Seq by RNA sequencing? It's gaining an increasing foothold in the sciences these days because it provides basically an unparalleled comprehensive analysis of the transcriptome. It addresses questions around quantification of gene expression, biomarker identification and discovery of novel transcripts, detection of fusion genes, as well as alternately spliced transcripts. And you get this all from, a single study rather than having to focus on, panels of individual transcripts at a time. So it's very, very powerful. But with that power and the plethora of data that it generates, so there are a number of challenges. It's both very, very labor intensive and lengthy. How accurate and reproducible are the results? It's a rather expensive proposition to get into. One also may question what kind of biases are in the read coverage. Was the full diversity of transcripts quantitatively captured from each sample? And then the other challenges that are presented in total RNA-seq is what the vast majority of total RNA is made up of sequences that are basically uninformative, whether these be ribosomal RNAs, or in the case of blood samples, sequences such as globin messages. So it's almost a prerequisite now to have some method that allows you to focus your reads from the workflow into actionable results. Now, you can get rid of ribosomal sequences through both depletion technologies as well as poly-A selection. But depletion technologies do bring a number of other advantages to the methodology, in that they retain non-polyadenylated transcripts. You can see here other long non-coding RNAs and other sequences that can be captured in addition to message. They enable detection of both long and short transcripts without any 3' bias. And that particularly becomes important when looking at degraded RNAs, where you won't have continuity in the transcripts between the poly-A tail and the remainder of the message. So depletion methodologies come in, and the problem with them is they can be rather lengthy and tedious to implement at the front end of a total RNA-seq workflow. Strandedness has become almost table stakes in terms of a requirement for total RNA-seq, because without strand specificity in the library, aligning the sequences and assignment of reads becomes very complicated, particularly if there are overlapping genes transcribed from opposing strands. In effect, with a strand-specific method for RNA-seq library prep, you in effect have kind of data-driven or an experimentally derived transcript annotation, whether it's for de novo assembly or for resequencing. Significantly improves variant analysis as well as resolution of otherwise sort of ambiguous data that may come out of your reads. So what we were looking at here, of course, with what we were planned to develop or tried to develop for RNA-seq library prep, okay? The central core of designing a system for the Illumina is basically locked. So you do have that black box, okay? And we can't do much about that. However, by focusing on optimizing the library prep process and streamlining this process for you, what we're hoping to offer out of this was basically better libraries in, better data out, and this is independent of whatever analysis pipeline you choose to apply to the back end or prefer to use or and have established for your lab currently. Now we can get into the SparQ RNA-Seq HMR Kit. Ultra fast, that may be a bit of a stretch in terms. These are, no matter how much we've tried to simplify these procedures, they still are somewhat time consuming, okay? I mean, and you MBAs out there, you can pick whatever kind of ameliorative, alliterative adjectives that you're choosing, I think, whether it's easy, efficient, and effective, rapid, robust, reliable, reproducible. This kit really has proven to meet all those kind of goals with this. But what we've done is we have definitely streamlined this process with this integrated ribosomal and globin depletion. The benefits that we're trying to bring to this and some of the properties is we've looked to minimize pipetting and transfer steps to reduce technical errors and bias that can be implemented or imposed by those actions. Kit consists of basically 3 essential reaction tubes, or there's 3 basic tubes or multi-steps that happen in those tubes, but 9 components and 9 simple steps to produce a very efficient ribosomal and globin depleted library with improved yields compared to others, leading methods and kits on the market. We see significantly enhanced reproducibility and uniformity across different amounts of RNA samples. And we also see an increased unique reads. So we're trying to both simplify the process and provide better results and reduce your time. So the kit whether or not you can actually get this done in a full 5 hours, most of the beta test feedback, they have been able to complete the process within a day, quantify the libraries, and have their libraries loaded on the sequencer that same day. And so we feel that does then definitely offer an advantage to the workflow. And we're trying to provide this to you in a more cost-efficient, very competitive price compared to other kits on the market with integrated depletion modules that are attached with their kits. So let's take a deeper look into the workflow, because this is where really the heart of the kit takes place. Again, 3 basic tubes where the reactions take place in. And let's take a look at each of these steps. So with the starting RNA, the kit's compatible with very good quality. Let's say of a RIN of 9 or greater down to at least a nanogram, a little more forgiving as you go to higher levels of input RNA from cellular, from cells, or universal reference RNA, let's say, high purity RNA. It's also very, very effective from 10 nanograms to 100 nanograms of degraded RNA. This may be FFPE RNA with a RIN in the range of, let's say, 3.4, 3.6. So the next step is where really most of the magic seems to happen with the kit. It's very complex, okay? So after adding the RNA, combining with the fragmentation and the priming buffer, we go through the depletion, the fragmentation. This is carried out essentially by heat with magnesium hydrolysis. The depletion happens during this step for both the ribosomal RNA and globin, and random primers anneal during this step. We then add the other enzymes required for first strand synthesis, carry out a short heat kill, and then move the reaction into second strand synthesis. And we are using essentially the industry standard or the field standard of uracil strand marking to carry out strand specificity for the process. Following this, there's a brief heat kill. I should mention that during second strand synthesis, there's also a DNA end polishing step. And so the DA tailing of the double-stranded cDNA happens during this step as well. And we go through a bead cleanup, classical ligation, 15 minutes with unique dual index Y adapters tuned for the sequence for the Illumina platforms with the I5 and I7 sequences. And then strand specificity with the DU strand marking, of course, occurs during the PCR enrichment step, wherein the master mix that we're using contains an archaeal thermostable DNA polymerase that does not copy, it does not recognize, it binds to the uracils in the second strand, and so the second strand is in effect not amplified, only the first strand is amplified. We follow that up with two times bead cleanup to produce a high purity double strand library ready for Illumina sequencing containing the P5, P7 sequences on either end. So again, generally, this can be carried out in roughly 5 hours of total time, reducing, I would say, total hands-on time by 30% compared to most other kits on the market with comparable workflows or technologies for strand-specific total RNA-seq. I think that's depicted more cleanly in this graphic. You can see that through this integration of the depletion into the first strand step, we're greatly simplifying the number of steps involved in this and reducing the total time compared to other leading brands on the market, and that have defined really the industry here for RNA-seq kits. So it's simple, it's quick. How effective is it? Well, if we look at RNA biotype representation, this is using universal human reference RNA, and we look at varying input quantities. Here we're looking at high input across the board of four different kits, starting with a microgram, and then looking at some of the lower input levels. We're showing 3 different input levels because kits from different suppliers have different kind of threshold limits that they recommend as being the minimal input quantity. And what jumps out is that the biotype representation, particularly in terms of protein coding reads that come out, is very consistent across a wide range of input of RNAs. And you also see that for more degraded RNA. Again, from FFPE RNA, you see very similar biotype profiles. Ribosomal RNA was detected at very, very low levels, comparable or better to other depletion modules that were employed with different kits on the market. And again, from blood, we have effective depletion of all globins. This includes both fetal and adult globin messages. So it provides a very flexible and widely deployable kit for most mammalian tissues. In terms of yield, we see here with the UHR from very low RNAs going across the board, we saw superior yield, at least in our assays. FFPE RNA, again, high yields. There were other kits that seemed to do fairly well here, but where things really stood out with FFPE RNA is when we looked at 5' to 3' coverage. Almost all the other major kits that we looked at, there was a rather profound breakdown and uneven coverage of sequence across reads. When looking at transcripts lengths from 2 to 5 kilobases, whether you had 10 nanograms or 25 nanograms of FFP RNA with the SparQ HMR kit, very consistent, very even sequence coverage from 5' to 3' N. And if you look at sort of a comparison of unique transcripts that were captured by the methods, again, you see very, very consistent coverage, very, very consistent metrics across a broad range of high purity, and as well as this being sustained in rather degraded FFPE RNA. And so one of the things that we wondered about was one metric that we saw when looking at some of the other kits on the market was there, and when you look at the base frequency obtained for each read position, they tended to cluster with the separation of seeing kind of a lower representation of G's and C's at each position compared to A's and T's. Whereas with the SparQ kit, and this was whether we had high input or low input, we saw very, very consistent, more tighter grouping, very even kind of sequence representation at each read position. And we questioned whether or not this would be reflected in the bias or coverage that we would see in the transcripts being captured by the process. And so we took a look at a panel of GC-rich transcripts. We took 10 just randomly selected GC-rich sequences and looked at total normalized transcript per million. So this normalizes the reads for length of each transcript. And you can see that we have very high sustained TPM for SparQ, whether we're looking at high input or whether we're looking at low input. Now, the flip side of this is if we're getting a greater coverage of GC-rich transcripts, has anything been compromised in the way of AT-rich transcripts? So we did a similar exercise and looked at a panel of 10 AT-rich genes. And again, you see higher TPMs for both high input and low inputs, arising from the SparQ. And our conclusion on this is that basically we have reduced sequence bias in libraries and improved transcript coverage. And that's been borne out by many of the external beta testers and early adopters of the kit that we've sent this out to. Generally, the feedback, we've had very, very positive feedback on both the workflow as well as the quality of the sequence metrics that these different researchers have obtained with the kit. So we're very, very excited and very encouraged by these data. And so just to summarize again, bringing back what we're hoping to accomplish with this product, we think it'll be very, very exciting addition to the Quanta product line. With a very simplified, fast, stranded RNA-seq library prep, providing this basically invisible globin and ribosomal depletion method, reducing pipetting steps, saving you time at the bench, still maintaining industry-leading metrics in what come in the libraries that come out, and providing this to you at a cost savings, and hoping to support and drive your research further. We do have a number of posters that probably get into a little more detail around this. I would encourage you to reach out to either sales reps, visit the QuantaBio website, download these, and you can inspect the data, peruse these, request a sample of the kit. And so we appreciate your time and feedback. Any questions you might have, feel free to drop me an email, reach out to me, dave.schuster@quantabio.com, or again, to any one of our sales reps. Be happy to address any questions you might have. Thanks.